This is my recorded presentation on prokaryotic cells and eukaryotic cells. We will be talking about the difference between these two types of cells. Now, what you're seeing on the left is actually a human cheek cell, and the darkened circle in the middle is the nucleus. Before I begin talking about the difference between two types of cells, I want to spend a few minutes talking about Henrietta Lacks. A lot of research that we do today on cellular biology is done on human cells that we grow in a petri dish. Now, all human cells that we use in research right now came from this one woman, Henrietta Lacks. My quote here, Black scientists and technicians, many of them women, use cells from a Black woman to help save the lives of millions of Americans, most of them white. And what I mean by save lives, or what the quote means, is that we have used our research on her cell line to develop different antibiotics and different treatments for diseases. So normally I have you watch the videos on your own, but I'm going to play this video in my recording because I think it is a very important look at how underrepresented groups have helped move science forward.
Henrietta Lacks was a sort of poor Black tobacco farmer who grew up in Southern Virginia. And in 1951, she was diagnosed with cervical cancer. At that point, she had moved up to Baltimore, and she went to Johns Hopkins for treatment because it was the only place anywhere near her that treated Black patients. So this was the era of segregation, and the hospitals had the quote unquote colored wards, which was the only place that Black patients or poor patients could be seen. And without telling her, before treating her tumor, her doctor just cut a little piece and put it in a dish. And he sent that down the hall to George Guy, who was the head of tissue culture research at Hopkins. And George Guy had been trying to grow human cells outside of the body for decades, and it had never worked. And for reasons that remain a mystery, Henrietta's cells just never died. So they started doubling their numbers every 24 hours. They went from like one dish to 2 to 4 to 8 to 16, and then just sort of pretty quickly took over the lab. And George Guy started calling his colleagues and saying, I think I have the first immortal human cell line, which is what they're called, which means they'll just grow and divide forever. And in response, his colleagues all said, great, can we have some? Because there'd been this enormous effort to grow cells outside the body for 100 years, because we really didn't know a lot about cells at that point. And in response to all of his colleagues asking for them, George Guy sent them to anyone who wanted to use them for research. And they spread around the world this way really fast. And they were one of the most important things that happened to medicine. They were used to help create the polio vaccine. They went up to the first space missions to see what would happen to human cells in zero gravity. They were used to create our most important cancer medications like vincristine and tamoxifen. Her cells were the first ever cloned, her genes some of the first ever mapped. They were used to help develop in vitro fertilization. Basically, almost all of the vaccines we take today can be traced back to research with her cells. There isn't a person out there who hasn't benefited from research on her cells.
The surprising tenacity of her cancer cells, which made them so important to medical research, also ultimately took her life. Henrietta Lacks died in October 1951, at the age of 31. As her cells were taken without her knowledge, in an era before the concept of informed consent existed, Henrietta died never knowing how important her cells would be. Even her family didn't learn of the true extent of her legacy until science writer Rebecca Skloot began working with Henrietta's daughter Deborah to uncover the truth almost 5 decades later.
Rebecca, she worked hand in hand with my mother, Deborah Lacks. They went on journeys to Clover, Virginia, to talk to family members. They did a lot of work together. They hit the road a lot. They put a lot of miles on their cars. My mom was 2 years old when Henrietta passed away. So she never knew her mother.
As Deborah learned about these things, there's so much mixed emotion in it, because on the one hand, look at all this incredible stuff my mother did for science and for the world. That gave her a level of peace with her mother's death that she had wanted her entire life. At the same time, there was a lot of anger about the fact that everyone has benefited from these cells except her family.
This information was out there for so many years, and the family was not even aware of Henrietta's, that they had taken cells from Henrietta Lacks, and these cells have been used all over the world by scientists, labs, researchers, and millions of dollars have been made, and you still have the family struggling with basic health care. You know, we got to recognize the past, but kind of like move forward and move in a positive direction to make sure that this doesn't happen again to our family or anybody else's family. And the Lacks family is making sure that we continue to move forward to honor and preserve her legacy and speak about her life.
So they collaborated with the NIH and formed what's known as the HeLa Genome Committee, where a few members of the Lacks family and a group of scientists sit on this committee, and anyone who wants to do research with the Henrietta's genome have to put in an application that is reviewed by members of the family and the NIH scientists. There's just, it was unprecedented. There's never been a moment like that where research, quote unquote, research subjects were a part of the decision-making mechanism of science.
The family now has a seat at the table. So instead of us being the last to know, we will be the first to know.
The immortal life of Henrietta Lacks is about so many different things. It's about science and communication and how important it is for scientists to be able to communicate with the public. It's a story about journalism, what happens when people tell your story. It's very much a story about race and science and race and medicine. So much of the history of science and medicine was built on the backs of Black people without their knowledge. And so it's so important that Henrietta's story be told as part of that history. And in some ways, that's the concept behind Black Lives Matter, is saying, yeah, this Black life mattered. And it matters that we talk about it, and it matters that we tell Black stories. And look at what can happen when we try to kind of squash those stories. It has generations of impact.
This year marks the 100th anniversary of Henrietta's birth, and her incredible immortal cells continue to be more important than ever. With the outbreak of the COVID-19 pandemic, the HeLa cells were at the forefront of the early research which helped to shape our understanding of the virus.
Within the first kind of few days of when we really realized, oh wow, this is a pandemic, this is terrifying, I started getting texts from members of the Lacks family saying, tell me Henrietta's got this. Like, HeLa cells are working on this, right? You know, and I do think that it says a lot about the Lacks family that their first response to the pandemic from so many of them was, okay, how is HeLa dealing with this? You know, we know she's got this.
It just makes me proud, because she's not only helping a specific group of people, but she's helping everybody worldwide. She's saving lives. She's giving life. So, I mean, yeah, with these, I say, magical cells.
When I do speaking engagements, people come up to me and they say, I had children because of your grandmother. That's a heartwarming feeling when, you know, your grandmother has died, but yet she's still providing life. So for her to know that she is saving and impacting so many lives, she would be smiling from ear to ear. That beautiful smile, that beautiful Henrietta smile, it'll light up a room.
As you watched this video, I think that it is so important to acknowledge the contributions of underrepresented groups to the advancement of science, but also to be aware that science has very much been dominated by white scientists and has benefited I guess, more of our white citizens. And so as you enter your careers and start to read more about science, I think that it is very important to be aware of these issues. So let's move on to look at the comparison of prokaryotic and eukaryotic cells. You need to explain the components of cell theory, identify 4 structures that all cells have in common, and then identify the 3 differences between prokaryotic and eukaryotic cells. There are actually more than 3 differences, but I'm going to highlight the ones I want you to know. Cell theory was developed in 1839 by Thomas Schwann. Now, prior to him coming up with cell theory, Robert Hooke in the late 1600s actually discovered cells. So he was looking at cork, which is part of bark, and he noticed these rectangular structures. Those are what plant cells look like. They look like rectangular structures. He thought they looked like cells or rooms, so he called them cells. Now, as microscopes got better and we started to discover more information about cells, that is when cell theory was developed in 1839. So all life is made of 1 or more cells. Now, notice that I say 1 or more cells. There is what we call unicellular life, and we've talked about this before. So living organisms like bacteria, unicellular means 1 cell. I'm going to put bacteria in parentheses. There is also multicellular life, more than 1 cell. And that is what you are probably most familiar with. The next component of cell theory is that the cell is the basic unit of life. So in a unicellular organism like bacteria, all the characteristics of life occur within that 1 cell. So I want you to think back on the characteristics of life. Reproduction. Well, bacteria can reproduce. It's not sexual reproduction. They reproduce with a form of asexual reproduction. Response to stimuli. Bacteria can respond to chemicals or proximity to other bacterial cells. So all of these characteristics of life can occur in a unicellular organism. Now, with a multicellular organism, the cells are more specialized. They can still reproduce and they can still carry out most of the characteristics of life. So that is why we call the cell the basic unit of life. Number 3, all cells come from earlier cells. If you think back on the origin of life on Earth, there was the original cell. It formed somewhat spontaneously. It was a very basic cell, but DNA developed in that cell, and that is the original cell. Now, all life today comes from earlier cells. Cells do not spontaneously generate. Since science has advanced and we have learned more about cells, we have added 3 more components to the cell theory, and we call this modern cell theory. Number 1, DNA is passed between cells during cell division. This occurs in multicellular and unicellular cell division. Think about a bacterial cell. You have 1 cell. In order for a bacterial cell to reproduce, it doubles its amount of DNA. These cells split apart, and now you have 2 bacterial cells. And that DNA is passed from one cell to another. You can't have a cell without DNA. Number two, cells of all organisms within a similar species are mostly the same. And when I say mostly the same, it refers to structurally and chemically. So the cell structures are mostly the same, and the chemical reactions occurring within the cell are mostly the same. And then finally, energy flow occurs within cells. Cells produce energy in the form of ATP, cellular energy. Let's move on to discussing which organisms are prokaryotic versus eukaryotic. Prokaryotic cells are bacteria and archaea. So what you are seeing in this image, these are the three domains of life. All living organisms fit into one of these domains. We have bacteria, archaea, and eukarya. All living organisms belong to one of these domains. Now, bacteria and archaea are what we call prokaryotic cells. I also will just refer to them as prokaryotes. Now, bacteria, like we've been talking about, and archaea are unicellular, one cell. Now, eukarya, the domain, I refer to all the organisms in eukarya. They can be called eukaryotes, or you can say eukaryotic cells. I'm just going to write eukaryotes. Now, this includes plants, animals, fungi, and a group that we call protists. You're probably not familiar with the term protist, but it includes single-celled organisms and multicellular organisms. So all prokaryotes have one cell. Eukaryotes can be single-celled or multicellular, because some protists are unicellular. This image shows you the four structures that all cells have in common. When I say all cells, I mean prokaryotes and eukaryotes. Now, in this image, you can only see a bacterial cell, which is a prokaryote, and plant in an animal cell. Those are eukaryotes. Now, all cells have a plasma membrane right here. In this bacterial cell, actually, the plasma membrane is the yellow structure in this particular image, right there. In a plant cell, the plasma membrane is this little strip right here. It occurs under the cell wall. And an animal cell, it is the blue structure that surrounds the cell. But all cells have a plasma membrane. Remember from your lessons on biological molecules that the plasma membrane is made of phospholipids. They belong to lipids. Now, all cells have a cytoplasm that is filled with cytosol. Some sources you read will just say cytoplasm, and that is fine. But I'm going to write the definition here for you. So cytoplasm refers to all of the space outside of the nucleus, which is that structure right here and right here. Now, I wrote here all the space that is located outside the nucleus or the condensed DNA. Now, the reason that I added condensed DNA is that prokaryotes, bacteria and archaea, do not have a nucleus. They still have DNA. And the DNA is this red structure you're seeing here. So the cytoplasm is the space all around that condensed DNA. In eukaryotes, it's the space that occurs all around the nucleus. Now, cytosol is inside of the cytoplasm. It's this jelly-like structure that includes chemicals or molecules like amino acids. So all cells, prokaryotes and eukaryotes, have a plasma membrane. That is a membrane that encircles the cell, and it is made of phospholipids. It lets certain molecules in and out and prevents others from flowing in and out. All cells have a cytoplasm or filled with cytosol. Number 3, all cells have DNA. You should know that DNA is the genetic material that you find in all living organisms. It is the genetic material found in all living organisms. Finally, all living organisms, prokaryotic and eukaryotic organisms, have ribosomes. Ribosomes produce or synthesize proteins. Produce and synthesize mean the same thing. So just to review, prokaryotic and eukaryotic cells have plasma membrane, cytoplasm, DNA, and ribosomes. So you need to know those 4 similarities between these 2 types of cells. This is a comparison of prokaryotic and eukaryotic cells. And when I say comparison, I mean the differences between these 2 types of cells. You need to know these 4 differences. There are other ones as well if you do reading online in your texts, but these are the 4 that I want you to know. Prokaryotic cells are small with a very simple structure. Eukaryotic cells are usually larger and they have a complex structure. So that means that there are more items inside of a eukaryotic cell. Number 2, a prokaryotic cell has what we call a nucleoid region, whereas a eukaryotic cell has a nucleus. So let's talk about the difference between these 2. And this applies to number 3 as well. A nucleus has a membrane around it, same as the phospholipid plasma membrane. It's a bunch of phospholipids and it forms a membrane that makes a compartment or enclosed area. So a nucleus is a membrane-bound compartment that holds the DNA. A membrane-bound compartment that holds the DNA. Prokaryotes, bacteria and archaea, do not have a nucleus. They do not have a membrane enclosed compartment for DNA. They have what we call a nucleoid region. What that means is the DNA is condensed or concentrated in one area of the cell. There is no compartment around a nucleoid region. Number 3 is a very easy way to tell a prokaryotic and eukaryotic cell apart. Prokaryotic cells do not have membrane-bound organelles, and eukaryotic cells do. When I say membrane-bound, again, I mean a phospholipid membrane. Now, organelles, think of them like little organs, right? In your body, you have a heart, a liver. In a cell, there are these compartments where specific processes are carried out. Those are membrane-bound organelles. An example of an organelle would be your mitochondria, and you'll learn about that in the next lecture. Within the mitochondria, ATP or cellular energy is produced. That is the function. Mitochondria has a membrane, and inside that compartment, ATP is produced. Well, prokaryotic cells do not have membrane-bound organelles. They still produce cellular energy, ATP, but they do not do it in a compartment. Finally, prokaryotes have a single circular DNA. So it actually looks like a circle. It is double-stranded, just like our DNA, but it forms a circle, whereas eukaryotes have linear chromosomes. We have linear structures, linear meaning straight structures that contain our genetic information. So we have multiple linear chromosomes, whereas prokaryotes have one circular piece of DNA. I guess you could call it a chromosome. I recommend that you watch this video I have put in here. I want to point out a few items in this drawing. This is a prokaryote and a eukaryote. That is an animal cell you are looking at for eukaryotes. Plant cells are rectangular and they have a cell wall, but animal cells do not. In this prokaryote, we have the nucleoid, condensed DNA. However, in the eukaryote, there is a membrane enclosed nucleus. Both cells have a cell membrane that circles the entire cell. Both cells have ribosomes. Those are structures that produce or synthesize proteins. And then eukaryotic cells have organelles, membrane-bound organelles. Those are all of the structures you see inside the cell. Notice that you do not see all of those structures in a prokaryotic cell. Also, prokaryotes are smaller and we call them simple, whereas eukaryotes are larger and complex. That summarizes the similarities and differences between these two types of cells. Now, I want you to make sure that you know which type of organisms fit into these two categories. Prokaryotes are bacteria and archaea. Eukaryotes are plant, animals, fungi, and protists. The next lecture you should watch after this is on cell structure in a eukaryotic cell.